And, okay, so we're live now with our Microbiology Journal Club. I have to laugh at myself because over the weekend I started, at the suggestion of Dave Baltris, I started putting links to our past journal clubs in PubMed Commons. And so because I had to do that, I ended up watching the very first 10 seconds of each of them because I wanted to make sure it was the right one. And I think I start every journal club going, okay, here we are. It's like, today I'm not going to do it, but I did. Oh, well. All right, so we're here to talk about this fantastic paper today, which is the latest in a series of papers out of Nicole King's lab, where she is looking at multicellular development in choanoflagellates and how bacteria impact that particular developmental process. So this came out earlier in 2016, so I think it's from the spring. And it was covered in Ed Yong's book, I Contain Multitudes. So as we were reading through that in the summer, I kind of thought about this as being an interesting topic. And then the paper kind of floated across my path again. And I thought, yeah, let's read this because I want to learn more about this particular system. It might be something that I should be including in symbiosis when I teach it again, because I didn't talk about it in this semester, but it certainly is something that we could incorporate. But before we get started, I'll introduce myself. I'm Laura Williams. I'm an assistant professor at Providence College in Providence, Rhode Island. I'm very delighted that the semester is done. It was a good semester, so I have no complaints about that, but it's always nice to kind of get to a break and get to recharge a little bit. And then Nicole, you're right next to me. Do you want to introduce yourself?
Yes, so I'm Nicole King, and I tweet as Old Dusty on the Twitter, and I'm a postdoctoral fellow at the University of Northern British Columbia in Prince George, where I've been studying bacterial and fungal responses to shallow excavation of soils. Basically dig a hole 20 centimeters deep and see what happens via ITS2 and 16S sequencing.
And Nicole has been very nice and has sent me a couple of really awesome emails that I'm working through because we're working on microbiome stuff and she's working on microbiome stuff. And as we're getting to microbiome data analysis, Nicole has been very helpful for that. So thank you so much.
Glad that you're finding it helpful. Awesome.
Very much so. And then Kat, would you like to introduce yourself?
I'm Kat Milligan-Myrie. I'm an assistant professor at the University of Alaska Anchorage, and I study how the host genetic background contributes to host microbe interactions using stickleback as a model.
And it's dark behind you. Is it dark behind you because it's dark out there in Alaska?
Happy solstice! Yeah, it's dark in Alaska. The walk today was beautiful. The sky was clear and there were stars just all around me and the moon was out and it was gorgeous. I did see a moose on my way to work, so life in Alaska.
Wow, no kidding. That's pretty cool. All right, that's very good. I don't do well with the short days, but the way you just described that sounds very beautiful. So I don't know.
Yeah, it's pretty amazing. I love my walks to work.
Excellent. That's excellent. Okay, so all right, so we are going to talk about choanoflagellates, which I admit I did not know a ton about before kind of reading the book over the summer and then getting into this paper a little bit. So if you guys know more about them than I do, feel free to correct me if I absolutely misstate something horribly. But I have to say, I really like her writing style. So I don't know if this is the product of the first author or the product of a collaborative effort between everybody, but I thought this was a really well-written paper that gets at the big ideas, but then also the details of what they're doing really well. So for people who are writing, you know, here's a good model for getting a good paper and getting your points across. So I thought what we'd start with is talking really briefly about choanoflagellates themselves. Because I think it's important to understand why they're working on this particular organism. So it's a protist, which I don't know if anybody's done any. Have either of you done work with protists before?
I have not worked on anything with a nucleus ever. I mean, other than the DNA from like fungi, so it's very much outside of my expertise. Yeah.
I studied Toxoplasma gondii for 7 years in graduate school. So
okay.
I've got some parasitology in my background.
Okay, great, great. I'm with Nicole. Like, if it's got a nucleus, I'm kind of like, that's so unnecessarily complicated. Why would you do that? But I feel bad for people who study protists because it really is this hugely diverse group of organisms, but it's just like this catch-all term that everybody goes, I don't know what to do with it. It's a protist, you know? And you'd lose, like, I think they get neglected. So
they really do. I'm just going to add to that I mean, especially working with soils, it's sort of thinking about, I mean, that's the other half of like thinking about the things that you do, Laura, and that I do as bacteria. I mean, somebody's eating them. Somebody's interacting with them. I mean, I've systematically been in studies where they're just being sort of not considered in some sorts of biological processes and spatially heterogeneous environments, considering that they can move around. You know, they've got flagella and they do twisty things, so they're pretty key, and they're still kind of a black box because I just haven't had a chance to look at them, and I suspect a lot of people haven't.
Yeah. I totally. Sorry.
Go ahead.
I totally agree. When I was in grad school, it was a constant struggle that we had to make people realize that our stuff mattered. It's a parasite, and Toxoplasma is even a disease-causing parasite. So you know, it has human consequences, and yet we still couldn't get people to get super excited. But now that everybody knows it's associated with schizophrenia, they're like, oh well, it's the one that makes your cats crazy and mice crazy, and oh, now it's interesting. But like, no, it was interesting before, people.
Yeah, I have great sympathy. Protistologists of the world, I have great sympathy for you because it is, I think it's, oh man, I mean, we could go off in a huge tangent about science funding and so forth, but like this is a group that you know, things like this, but other applications, other practical understandings, other theoretical understandings, there's a lot of work to do in protists, and the protists are a huge, huge group. So I'm sad that they're neglected.
I just wanted to add to that. I didn't think about coagulates, I cannot say the word, much until I heard Rosie Elgado's talk at ASM Beneficial Microbes this fall. And she just made it sound like this, it's this evolutionary model where we can study these single microbes that come together because of a signal from bacteria. And that just got me super excited. And I was like, wait a sec. I mean, I knew protists were exciting because I studied them, but all of a sudden I'm like, wait, this is an evolutionary model that we're not really taking advantage of. And of course, people are, because the King group has been doing this for a while, and they recognize how, what a wonderfully complex and interesting system this is. But I had not considered it. And after hearing Rosie's talk, I was like, everybody should be studying this. Everybody in evolution needs, or many people, we need more people studying this in evolution and in host microbe studies, because it's just a wonderful system where you can manipulate the bacteria, and you can manipulate the host, and you can manipulate the environment so well. So I just, ever since Rosie's talk, I've just been really impressed with the system.
Yeah, absolutely. I think that was very well said. Much more eloquently than I think I would have managed that. But it's all the points that, and they do lay this out in the introduction incredibly well. So I would say if you're somebody who's not familiar with this system with choanoflagellates, this is a good paper to read, because the introduction very quickly but very clearly lays out, like, bing, 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 here are the reasons why this is a great system. And they're all the reasons that you mentioned. So I did want to show, oh, dangerous. I'm going to see if I can actually do my share screen thing. Haha. I doubt it. Aha, is it going to work? Yay. There we go. Let's do this. So I just wanted to quickly show, can you guys see the little image there? No? Present to everyone. Let's try that. Good? All right. So I just wanted to show this just for people who might not be familiar with it. This is just from Wikimedia Commons. So this is a little picture of a choanoflagellate. And so one of the things that they talk about is this, now if I'm getting this wrong, just gesture wildly at me. But the little collar here is kind of one of the things they talk about in the introduction, is that they have this distinctive collar of actin-filled microvilli that surrounds this flow-generating apical flagellum, which you can kind of see at the top there. And so what they do is they basically, it sounds to me like what they're doing is they're kind of sweeping prey bacteria into this opening as a means of getting nutrients, basically. So they're predators on the bacteria that they find in their environment. And they draw this really excellent, it's just one sentence, but I thought, oh, that's so true. They draw this great comparison between this microvilli in the collar and you know, the animal epithelial tissue. So in the gut, for instance, that you've got these epithelial layers with microvilli that are going to be the primary point of contact with our gut bacteria. So just like Kat was saying, this is a great system because it has some parallels with things that we are interested in terms of understanding how our tissues and our systems are interacting with bacteria. But it's so complicated to do this in humans because we have this vast gut microbiome that's transient and changing and different for all of these reasons. But in this system, you can really bring it into the lab and exercise a really detailed level of control over who's there, what are they being fed, you know, when are they being fed, all the interactions. So it's really exciting that they can do that with this. Okay, so I was also going to show you, in terms of multicellular development, this, if it'll work, there we go, this little picture, which is, it's not the same genus that they're working with in this one, but it is a choanoflagellate, and this is the colonial form. So this is the rosette that they're talking about. So the key thing here is that it will transition from a single-celled stage into this multicellular stage. And they make a point to say this is not just through cell aggregation. So this isn't like cells are swarming together. But what this really is one cell goes through a divisional process, and those progeny cells remain together, and they end up forming this kind of colonial form, which, you know, if you think about it, that's very much what, you know, if you're thinking about embryonic development, if you're thinking about all of that, you're like, oh, yep, okay, this is sounding very familiar. And so the key thing they're looking at here is the cues from environmental bacteria that lead to the transition from that single-celled state into that multicellular rosette. And they talk a little bit, and they get back to this in the discussion, but they talk a little bit about the fact that it's a fitness advantage. I don't know, did you guys have thoughts about, because I hadn't really thought about what would be the selective pressure for this, but certainly that is a very key question about why is this interaction this way. Did you guys have any thoughts about their hypotheses about why this would be a fitness advantage?
I think the idea about everybody getting to eat more is that's sound to me. I mean, this is really interesting because, I mean, you have like these cells with this sort of morphological, you know, attribute. Number one, having villi and a collar, not just to create current, but I mean, they're all getting together and they all have this sort of maximized region of surface area, but also it is kind of creating a bit of a microenvironment, even if it's just currents to bring enough prey in. So the idea that you might just have these rosettes that are probably dividing more efficiently, probably cells with better nutrients, better gene expression in general. I mean, it really does make sense from a proliferative standpoint that they would outcompete cells that were perhaps more shear sensitive while trying to form rosettes. So I totally think, and I mean, particularly if this is happening in aquatic environments where you've already got inherent shear and inherent fluid flow, I mean, they're probably going to be able to stay their ground, be big enough to hide behind a crevice somewhere and not be swept away while they're trying to do this business of eat, reproduce, and go about all the biochemical and biological things that you do. So I think I have no problem with that speculation, really.
I think it's fun thinking about how you would test that. And I agree. I think I totally agree with Nicole that there are probably advantages to living in this area, living together where you can prevent cells from floating off individually. And if you find a good food source, then this is a great way to be able to concentrate around that food source. But I think it'd be really fun to test this in the laboratory and thinking about ideas like, would you set up a flow system to see if they're able to be more stable in this environment? How would it help with prey? You know, it'd be fun to be able to see if, because they talk in the very beginning about predator prey, and wouldn't it be interesting to see if, you know, if you have a predator coming in, do they scatter, and then or do they stay together because as a unit they're able to look bigger to this prey, so maybe predator, so the predator is less likely to attack them. Or yeah, I just think that there are some really fun ways to be able to test this.
Yeah, that's a good point. I didn't actually think about the, you know, we're thinking about the bacteria, and then the bacteria are being preyed upon by the choanoflagellates, but I actually didn't think about the fact that, of course, there's going to be another level, that there's something that's going to potentially be eating the choanoflagellates too. So that's a really good point. I hadn't thought about that as being an aspect of the rosette formation as well. Okay, so we got these rosettes that are forming, and past work from the King lab demonstrated that it seems that the initiation of rosette development, so this developmental pattern, appears to be cued by the presence of an environmental bacterium called, here we go, Algoraphagus machiponganensis. I had to look that up last night. It's like, oh man, come on. So it's this particular environmental bacterium that provides the cues for the transition from the single-celled state to the multicellular rosette state. And they knew that already. And going into this paper, they'd identified something called the rosette-inducing factor 1, or RIF1, which is a particular kind of a, let's see, I always get my lipid names mixed up. But this is this the one that's it's a bioactive lipid, but they do talk about it being a specific kind of lipid. And now I'm not going to be able to find it.
I'm just unmuting. It's a sulfonolipid, I believe.
There you go. Thank you. I was going to say, for some reason, sphingolipid was in my head, and I'm like, that's not right. It's a sulfonolipid. Okay. All right. So, and what they wanted to do was they wanted to figure out, are there other bioactive molecules being produced by these bacteria? Because what they noticed was if they just have RIF1, that doesn't induce the same level of rosette formation as if they're exposed to the live bacteria. So they thought, clearly there's something else going on. Let's see if we could figure out, there appears to be a combination of molecules that are impacting this. So in addition to the just one, let's see if we can find the other ones. And so that's really the major goal of this particular paper. And let me really quickly, I'm going to do my screen share thing. So for some reason this doesn't work, let me know. But I think this kind of helps to get that. All right, so hopefully you guys can see the panels there. So figure 2A, that was just a nice kind of, they're just setting up, they're looking at rosette development. So a lot of the things on the y-axis are going to be percentage of cells in rosettes, which is a nice, in terms of having a bioassay, that's a nice simple one to be able to do. You're just like, let's count the cells in rosettes. Ta-da! So what they've got there is, go ahead, Kat.
I just want to say, working at a PUI, a primary undergrad institute, I was really, I thought, what a great system where you could, you know, get these images and then have students work on counting how many are in a group, and what a great way to engage undergrads. Or I also thought that maybe this would be a fun system to have in the lab, you know, where we talk about how to get students more excited about microorganisms, and this would be a great way to get students interested in protozoa, because, or flagellates. I almost said it right. Because you could set something up like this in the lab, and I think it would be a really fun way for students to create their own experiments. It's something that they could do relatively easily and quickly. So I thought it was a great system.
Yeah, absolutely. That's a really good point. And it's certainly true that when you could give, or at least from my brief experience with this, when you've got this very clear, I mean, this is, to be able to count the cells, so you've taken the data, you can count the cells, but to be able to tie that immediately to once you get these numbers of cells and you can plot this, that's what this is going to tell us about how these different things impact multicellular development. I mean, to connect that so quickly from you counted these cells, that's what this tells us about our big question, that's great. Because I think students get frustrated when they go through all this kind of this work of counting the cells and they're like, I don't know what this means or why we've done this at all. So yeah, I could see this being a really good system. And you've just given me a good idea about thinking about maybe we could develop this for a Gen Bio lab or something like that. I think that could be really good.
Yeah, one of the things I like about it is you can tie in both microbiology, host microbe interactions, and evolution in one system. And that'd be a great way for students to think about how does, one, early evolution, which is something we talked about early on, but then two, host microbe interactions. And often when we talk about that, we talk about larger systems you can't take apart. It's hard to take apart an intestine and look at how one microbe is interacting with another microbe in a timely manner where students can do it in a lab period. But this, I mean, I don't know exactly how long this would take. It sounds like they expose the God, I cannot say this word, coagulate, that one, to the bacteria for 24 or 48 hours, and then they look at and then they quantify the rosettes. But that sounds like something where you could have the students do it on Monday, and then on Wednesday come in and get the results and plot them out. So it just seems like a great way to not just look at microbes and not just look at host microbe interactions, but host microbe interactions and evolution. And maybe some biochemistry if they are able to get their own fractions. Go ahead, Nicole.
I was just going to add to that, like, the beauty of this whole paper is how additive and subtractive the entire approach was. And even thinking about this for undergrads, which I hadn't really thought about because I do less teaching, but I mean, I did not read the methods except for wanting to know how much they were using for chromatography. But even if you just wanted to have a lab as simple as live bacteria and conditioned media, all you need is a centrifuge to actually look at the two parts, which is very nice, actually, to be able to show students that, yes, these are secreted factors and not just cells that are mediating this, this behavior of the Kawano. So, I mean, there's a lot of things you can do here from the classroom standpoint that aren't going to break the bank and that aren't going to require having to bring in fancy things just to introduce one or two concepts. I agree. It is a very exciting system to use that way.
Yeah, I think so too. So I'm gonna, I might harass our Gen Bio lab coordinator because he is in fact looking for lab exercises for this upcoming spring. So I might send him this paper and say, hey there. He's visiting family right now, so he has something, he needs something to fill his time. So I could like send him this paper and say, let's do this. Yeah, that's a great idea. I think there's a lot of things you could do with that to help students make these broad connections, but with something that is not incredibly intricate or complicated. So yeah, so what they're showing here is they've got percentage of cells in rosettes on the y-axis, and then in 2A, because I'm never sure if anybody could see my arrow, in 2A, what they're just showing is basically this is kind of the figure of what they knew already. So, they don't form rosettes in a media control. And I know from reading Ed Yong's book that one little media control category and all those little dots at 0 was, I think, years of frustration for the lab trying to figure out why they couldn't get them to form rosettes in the lab. So it's funny when all your sad saga of scientific frustration can get squashed down into like one part of a graph. But they did figure it out eventually that they need this cue from bacteria. So what they're showing in 2A is the live bacteria, conditioned medium, outer membrane vesicles, and then the lipid extract. They're kind of narrowing it down to show you that these lipids that are being produced by the bacteria are the cue that leads to rosette production. So you get about the same level of rosette production with all 4 of those. And then what I'm going to do is I'm going to actually skip over B because I think it might be easier to talk about C first, because what they basically did was they said, okay, we want to look at the lipids. And so what we're going to do is we're going to do high performance liquid chromatography, which I've never done, but we'll put your lipid extract into fractions. So it's a way to try to take all of your lipids and start to split them up into groups so that you can then look at the activity of individual fractions. And they already knew about RIF1. And what they found for 2C was here they're actually looking at two lipids that they found in fraction 11. So for the moment, we're going to ignore 2B because I think that gets into the combinatorial stuff that's a little bit confusing at the moment. But for C, what they did was they said, aha, we actually managed to find something else, which is what they're going to call RIF2. So what C is showing is the solid squares are the RIF mix. So I think that's just fraction 11 altogether. And then they purified RIF2, which is the solid circles. And they found that this RIF2, in fact, induced more rosette development than the RIF1 that they knew about before. Because you guys can correct me if I'm wrong, but I think what the reason for the inset is because RIF1 really only gets to about 1 to 1.5% of the cells in a rosette. So that's why they've got that tiny little gray thing at the bottom. Is that how you guys read that too?
Yes, that's pretty much what I got. I mean, they do actually introduce these relative activations rather articulately in the results section too, to go with this figure, particularly as they sort of pick apart the RIF1 versus RIF2 rosette induction.
Yeah, so I actually don't. Hey, Dave, how are you? So I actually don't. So we pretty much just started into figure 2. We were expounding the virtues of choanoflagellates as an awesome system for doing experiments with students. And so now we're kind of, I don't know the history of how they found RIF1, but given that it has such low activity in terms of inducing rosette development, I'm kind of impressed that that's the first one that they found, because it seems like they might have encountered RIF2 first, because it certainly is more active. One of the things they do point out, though, is that, as you can see from the graph, the fraction 11, or that RIF mix, because it's the higher one, you still get more cells in rosettes. So RIF1 plus RIF2, if you add those two together, that doesn't cut it. So they're basically, their outcome from that is to say, well, it's clear that RIF2 is important for rosette development, but it's certainly not the only determinant, because the mix together, there must be something even within that mix that we still haven't figured out, because the mix will get over 20% of cells in rosettes, whereas RIF2 by itself only gets you about 10%, and RIF1 gets you kind of a measly 1 to 1.5%. So I think that might be what led them to start looking at combinations of fractions. So that gets us back to 2B. So in 2B, what they're looking at is, if they combine lipid fractions together, do they see synergistic effects or do they see inhibitory effects? My impression from this is that they expected to only see synergistic effects, but they were kind of surprised to find that there was a fraction that combined with fraction 11 gave them inhibition. And so that is the 4 and 5 if you're looking at that panel 2B, because the darker colors mean fewer cells in rosettes, the lighter colors mean more cells in rosettes. And you could see 11 is kind of humming along, and then it gets combined with 4 and 5, and your percent of cells in rosettes crashes. And then if you combine it with 7, you get more rosettes. Hopefully that was the outcome that you were supposed to get from that, but I think that's true. So then they said, all right, so let's kind of try to figure out what's going on. I think they started by trying to figure out what's going on with the inhibition. And so here is where somebody who has a better grip on chemistry than my brain does right now can help me answer a question, which is, so RIF1 and RIF2, both those chains are part of the same molecule, yes?
Yes, they are.
Okay. So you have got the inhibitory compound, which is this IOR1, is missing that fatty acid, is that correct?
Yeah, so the amide linked chain at the top is not part of IOR1.
Okay, got it. So it shares kind of that, what did they call it? The capnoid base. So it shares that capnoid base structure, but it's missing that fatty acid chain.
Mm-hmm.
Got it. Okay. And so then I think the thinking is that that's the reason why IOR1 is an inhibitor. Is that correct? Because it shares enough structural similarity that it's going to be able to bind to some mystery receptor that they don't know about yet.
Yeah, and this is the part, that I think is really interesting, because you can see that there's enough structural similarity here that this entire set of plurality, and there probably is a plurality of regulatory responses as this set of cells, starts aggregating together in response to these bacterial secreted components, that, they're probably all doing some degree of interplay at a binding site that likes long floppy carbon chains, but to different extents. So is presence or absence of an upper chain changing what the signaling is for one or multiple receptors? It's pretty fascinating, but yes, I mean, the structural similarity with IOR1 there to the sulfano moiety possessing lower chains of everything else does suggest that they're all competing for binding to, I think, the same response regulator. We're strongly suggestive of that.
Yeah, that makes sense to me.
Yeah.
So then so the B, sulfobasin, I'm not sure, that is an inactive compound that's produced by Algoriphagus. So it's a sulfonolipid, but they are calling it an inactive one, which I interpreted to mean it doesn't have any activity with inducing rosettes. And I have to admit, I did read this at like 10 o'clock last night, but I stared at it. I was like, it looks the same as everybody else. Like, why is it not?
Yeah, there's a hydroxyl group.
Yeah.
Where am I missing the important key piece? Oh, is it missing the hydroxyl group?
Yeah, it's missing the hydroxyl group from RIF1, and then it's missing the double bond from RIF2. And I thought that was kind of cool, because here you have this molecule that looks so similar to RIF1 and RIF2 and has none of the activity. I just thought that was, I think biochemistry is fascinating, because how would you think about a molecule, or you think about a cell that has to be able to differentiate between RIF1, RIF2, and sulfobasin, whatever, F, and it's going to have a very different reaction to each one, and the only thing different is that hydroxyl group and the double bond on carbon 5. That's just pretty amazing.
Yeah, whenever we hit things like this in classes, when we're reading primary literature, I always call this, for my biology students, revenge of the chemists. Because they take chemistry and they're like, chemistry is so hard. When am I ever going to use it? And then you just point at this and you go, it's revenge of the chemists, you guys. You have to know this stuff. They end up being important reasons why chemical structures, and as you guys just pointed out, like, it's one hydroxyl group and a double bond, really. It's going to be making the difference here between an active compound and an inactive compound.
Yeah,
it's also not that they're probably kind of jumping.
Oh, Dave, you're going in and out.
My internet sucks today, so I might. Yeah. I need Joel's like figure thingy.
Yeah, you just draw it out.
It's probably jumping both of them, right? Yeah. I'm just gonna observe, because apparently everybody is downloading everything today in Tucson. So.
Okay. Nicole, were you gonna say something?
Yeah, I mean, the chemistry on RIF2. I mean, I've gotten rusty since my PhD, but I mean, it does look to me like if you did somehow manage to get deprotonation at the red OH, that you could have some delocalization of that double bond as well, which would be rather interesting if the whole reason that RIF2, RIF1, and IOR1 are binding to this response regulator is because you can actually get the mobility that you'd have of no double bond in that lower chain. I mean, double bonds do make long chains like that more conformationally rigid. So if there's some sort of pre-processing of RIF2 before it's actually doing things, would also be kind of neat, But I mean, I also just appreciate the methodology here, because I know in the supplementary And this is also why I envy people who are working on cultivation-dependent stuff, is that they could grow 15 liters of Alcor, of A. matripongensis to get it through a column and actually look at all of these fabulous responses. Although I feel like the technician or undergrad or grad student who did this probably never wants to see a liquid chromatography system again in their lives.
Probably so.
Yeah.
Dave's got a sign up. He's saying pH.
Yes.
Dave says pH.
Well,
so,
yeah. I mean,
he's like, now interpret that as you may. Yeah. So yeah, that's interesting. So you've got all of this. So I think what Dave was trying to say with this cue card is that this is you were saying you're gonna flip back and forth based on things like environmental conditions and pH and so forth. Aha, there we go. All right. So that's why you've got this range of compounds. You've got slightly different chemical structures. You're going to get different recognition. That's going to be impacted by things like environmental conditions, pH, so forth. But yeah, no, this is, again, in terms of a system, as we were talking about earlier, in terms of a system to try to help students really grasp a lot of concepts at the same time, now you've brought in chemistry too, which is to emphasize to them, like, you don't study chemistry in isolation from biology. You really need to think about all of this together. And I think I'm gonna wait until the discussion to get back to this. But so one of the things that they're showing here is when you combine fractions 4 and 5, which they identified as containing this inhibitor, with 11, so you're seeing an inhibition of the RIF mix that they had already identified in 11. So once we kind of get the whole system, because there's one component we haven't talked about, one of the things I am going to ask you guys is, what would be the purpose of having an inhibitor? So keep that in mind, because we'll come back to that, because that was one of my big questions at the end of all of this. But in addition to identifying an inhibitor, they also identified an enhancer, so a synergistic compound. And that is shown, I mean, that just jumps out at you with 2B. That fraction 11, which is the one that contains the RIF mix that they kind of already knew about. But once they put it with fraction 7, you just got it shot up in terms of the proportion of cells that were in rosettes. So they said, obviously, we've got to take a look at fraction 7, figure out what's going on there. And what they found, hopefully this doesn't make anybody dizzy, but what they found was this, which are, here we go, lysophosphatidylethanolamines. What? Said it. Yay. Ooh, I see your note, Dave. Okay, so this the LPEs, because that is the last time I'm going to say that whole name through this journal club. So the LPEs they've got here, so they're looking at 2 different ones that they've identified, and then down here, what they looked at was combining the LPEs with the sulfonolipid RIFs to try to figure out, do they have a synergistic effect on rosette development? So the way I saw this was they have got, on the x-axis, they varied the concentration of the sulfonolipid, and the way I understood this is that they kept the same concentration of this LPE that they have just discovered. So I think in this graph, it was always 2 micromolar LPE, and then they combined it with either RIF2 on its own, which is the circles, or the RIF mix, which we know has RIF1, RIF2, probably some other stuff too. Those are the squares. And so what you can see is the closed circles and squares is without LPE, but if you add LPE, you get this additive effect where you're seeing lots more cells in rosettes because of the combined action. And so even if all you've got is RIF2, you're seeing that kind of shoot up once you add these LPEs. And I think it's the 2 of them together, so it's an LPE mix. So what they're finding there is you've got multiple components being produced. And I don't think they really expected that. So I think this paper kind of is the first time they realized that this particular type of lipid is going to be having this activity. Anybody have any thoughts on this one?
I'm just going to unmute and say that I think because the polar head group that phosphate with the amine chain, as well as the ester linkage in there, to me, this is actually suggesting that there's probably two types of ligand binding molecules that on the Kawano that are probably at play in regulating this rosette formation. So this is me being bold and guessing, but like I don't really think that they're all. I think everything in Figure 3, particularly IR1, RIF2, and RIF1, are doing some interesting things for at a single or a dual binding site on one response regulator. And I'm using that term loosely. I don't mean it in the way that quorum sensing people use it. It's just whatever's leading to rosette formation. And just because of the LPEs with the charge distribution, that there's something else going on. It might be triggering something about cellular attachment that's more synergistic for this. Maybe it's activating some idea of sensing or aggregating or cell division with those choanos. But I feel like there's two proteins at play to receive these bacterial originating small molecules. And it's actually really cool that they're, you know, long hydrophobic things to me. So I kind of wonder why that's the case, like why it's these sorts of molecules in particular, but that's just me musing sort of aimlessly. I feel like this paper is the envy of most, well, I don't know. I mean, I've been to conferences with natural products chemists, and some of them ooh and ah over structure forever, but like, this study is so gorgeous just because everything's getting linked to like this end, you know, multicellular morphology, and I'm just kind of enjoying the walkthrough with it, honestly.
Yeah, and I think it's always nice when you know, because just in the way we were talking about earlier, that you have people who study protists who are just like always desperately trying to get people to appreciate why it's good to know about protists. Again, you've got chemists who are basically just like, please understand why this structure is important. That double bond right there is key, I promise. And everyone's like, oh, I don't know. So it's good to, as you said, you're right. This is really nice to be able to kind of link these chemical structures, the variations that you're seeing in ways that you can actually see the outcome of it in this big biological concept that you're looking at. Dave just pointed out it's nice to be an HHMI funded investigator. So I think that is a factor. It's true. Okay. So one of the things that they wanted to do then, and I'm going to go to this, all right, I'm going to go to this violin box plot. I think those look more like cellos, but you know, we're not going to argue about that. That's a cello box plot to me. All right, so what they wanted to do now is they wanted to start kind of understanding what do you think the role of these LPEs are. And so they're developing a hypothesis here that they're going to argue that it has to do with maturation of the rosette. So if I interpreted this right, my understanding was they don't think the LPEs, I think they felt like maybe it doesn't have as much of a role in inducing the rosette development than it does to kind of helping the rosette kind of mature, hold together, keep the structure that they're kind of used to seeing in a wild type rosette. Is that kind of consistent with the read that you guys had of it? Okay, Kat's nodding. All right, so here's what we've got. What they're showing you in this violin box plot is they've got outer membrane vesicles on the left. That's their positive control. So that's basically like saying all of the lipids that Algoraphagus is going to produce should be in these outer membrane vesicles. So any compound that has activity for rosette development should be in these outer membrane vesicles. That's our positive control. And then they just looked at RIF2 on its own, which they know induces rosette development, but they wanted to kind of understand, what do the rosettes look like? You know, what are they structurally? How do they hold together? Are they resistant to shear forces if you just have RIF2? And then they looked at the same idea, RIF2 plus these newly discovered LPEs. And on the y-axis, what they're showing here is basically number of cells per rosette. So now this is where my students always get confused when we do primary literature, is because of the way that they show statistical significance up there at the top. Like if you've read enough of these papers, you're like, okay, I understand what they're saying. But every time I go through primary literature with students, they've read the text and they say, oh, well, that figure shows us this. Like, that's true, but what is the statistical test that they did? What are they comparing? How are they showing significance? And they usually stare at those little bars and asterisks and stuff, and they go, I don't actually know. So I always make them go through this. But basically what they're doing is they're just doing pairwise comparisons between the groups. So they're looking at, is there any difference, there's between outer membrane vesicles and RIF2, we see a drop-off in terms of number of cells per rosette. Between RIF2 and LPEs and RIF2 alone, you see a drop-off, a statistically significant drop-off in terms of numbers of cells per rosette. But if you're looking the top bar, if you're going to compare outer membrane vesicles and RIF2 plus LPEs, they did not see a statistically significant difference. So it looks like RIF2 and the LPEs is recapitulating a lot of what you see if you have just outer membrane vesicles. I think one of the things I wanted to ask you guys was this frequency distribution. So they picked this violent box plot on purpose to do this frequency distribution. When I was looking at it, though, I wasn't entirely sure. What do you think is coming out of that? Is there something from that frequency distribution that we should be picking up on that's telling us something about RIF2, LPEs, and the other compounds that Algoraphagus is producing?
One of the things
go ahead. Go ahead. It's all good.
One of the things I find interesting about these is that, when we show, one of the reasons I actually like this kind of plot compared to other like bar charts or something, is that when you do a bar chart, you would miss the fact that there are a lot of rosettes or a lot of cells that aren't in huge rosettes, and that there's a small number of really big rosettes, and then a large number of rosettes that are just between the 6 and or 4 to 10 cell stage. And so that's interesting to me, because I imagine, I'm assuming that these are in a relatively mixed solution, and so the fact that there would be some cells that are forming these really nice tight rosettes, and then a lot of them that are forming kind of smaller rosettes is interesting to me, and it makes me wonder if that inhibitory factor maybe becomes activated at or if there's something that in the protozoa cells themselves that are actually not protozoa, but in the choanoflagellates, get that word, if there's something when they get up to a really large cell stage like that they are actually inhibiting binding of the of these molecules, the RIF2 and the LPE mix, when they get to these really large molecules. And then I just saw Dave's comment, maybe they all go to 11 if you give them enough time. Okay. Oh, I see what you're saying. So maybe it's a time thing, but I see what you're saying. But yeah, I think this type of plot gives you a more accurate reflection of how many of these really large rosettes are forming, and it turns out there's not a ton of them, but when you have just RIF2 alone, there are none of them. So there's this I like the fact that you can actually see this distribution, and I think it's interesting that there's this decrease between 10 and 16, and it makes me wonder if it's a spatial thing or if there's a signal within the rosette itself that's turning off the ability to form even larger rosettes.
Yeah, Nicole, did you have something you wanted to mention?
Yeah, and this is going to be a partially, my understanding of statistics is really not great. I'm sure there's still time to learn, but my understanding of how how these violin plots get plotted is that the shapes that resemble cellos, or I actually think they kind of look like the snot suckers that you give to babies, at least the two on the ends, and then the one in the middle just looks like a flattened syringe. They're called in some of the R literature probability density kernels, which I don't get, but I think the thing that I really like about these plots, actually, if you're thinking about, let's say we're in the engineering mindset and doing like, how well are we mimicking our biological system? So we know that OMV is kind of like the best case, aside from having full-on A matropongensis. The fact that you actually have a slimmer but still recognizable snot sucker distribution on your far right, it's kind of nice that you can actually use this variation in sort of the distribution of rosettes that you're getting from that shape to let you know we're really close to simulating, you know, the biological process of rosette formation based on this distribution with the parts we've added in, whereas with the RIF2 alone, it's very clear that you're getting, you know, a combinatorially something else going on with your cells that isn't quite, you know, what you want on the left side. And I think that's also, I guess, an interesting diagnostic component to this plot. And I think that's really the only comment I had, because I don't tend to use violin plots a lot in my data, in part because of the low replication, but also because of not really, you know, looking at things that would have this much heterogeneity in what you get back. But I think it's certainly a very good choice of visualization in this case, and in combination with this assay.
Yeah, I absolutely agree. When I saw this plot, I thought, oh, that's such a great idea. So Dave is saying the RIF2 plot is differently shaped because axis is inherently truncated. Oh, I guess what, so everybody's getting smashed down because they've got 4. It's the same shape if you spread it out. Oh, I see what you're saying. So if you were to expand from 4 to 8, you might get that same snot sucker looking shape, but it's stretched out for the other 2, whereas your upper limit for RIF2 appears to be 8. So from 4 to 8, if you could stretch that out, you might get the same, I think Dave's saying you might get the same general shape. But I think for RIF2, the bottom is pretty squashed. But I agree. I think these plots are really great for showing the variation in terms of what, because someone pointed, I think it was Kat that pointed out that kind of it's a little narrower for RIF2 plus LPEs at the top, and then the bulb part between 4 and 8 is a little broader. And I think one of the things that Dave was saying was what you may not know here is the impact of time. So is this just because you cut all of these off at the same time and that's as far as they'd gotten in the one on the very right? So if you'd given them more time, then you could maybe get them to, maybe it would start to approximate the OMV's shape a little bit more even. And then Dave is saying would be good to show the percent of cells in rosettes versus the percent of single cells. That isn't what they're showing here. That had been what they were showing before on y-axis, but so RIF2 might be signal to assemble, other might be to assemble bigger. That's true, yeah. That's true. So I think that feeds into what they were saying, that maybe RIF2 is active in inducing rosette formation, and then the LPEs, which you could see there, is the effect of them actually maturing, like you typically see with the wild type lipids. Yeah, that's really interesting. I agree with you, Nicole. I think that was a good way to try to capture the biological phenomenon beyond just showing the median and the range.
It's also a lot nicer than throwing another dispersion test on for that data so that there's another column of numbers to look at. So I think it was wisely chosen. Yeah,
I agree. Kat, did you have something you wanted to mention?
Yeah, I wanted to bring up a couple of things. So one of the reasons that RIF2 looks so flat on the bottom is because they're not actually showing the doubles and the singles, which is, like you said, something that they were showing before, that they were showing the percentage of cells that weren't in rosettes at all. So I thought that was interesting. And then the second point that you made about the organization, that's one of the things I like about the system, is that one of the things that they discuss in the results and in the discussion is that they're not just looking at molecules that perhaps bring these individual cells together, but possibly holding them tighter together. And that's something that we would think about in Toxoplasma. And it turned out that in Toxoplasma, there are actually individual organelles that are responsible for releasing some molecules that just bring the cells together, and then other organelles that are responsible for releasing molecules that help you get into the cell, and then another organelle that helps you form a parasitophilous vacuole. So it'd be fun to see where these specific lipids and RIF factors are actually binding to in the cell, and it'll be really interesting to see as they're developing developing a system, to see which molecules are responding and where they're responding, what part of the cell those molecules are coming from, because I'm going to guess that the lipids, activate this pathway, and there's all these downward things, and that eventually you have something inside the cell, and it'd be fun to see if there's a specific organelle that's driving the release of molecules that drive the ability of those rosettes to stay more tightly packed, and that's what they're showing in in my version, it's actually figure 5. in 5E is that those tightly packed rosettes in the RIF2 plus LPE versus the RIF2 alone.
Yeah, I think that I looked at the clock to realize we're getting close to 2 o'clock. So I try not to keep people. No. I try not to keep people past an hour. But I was going to say that, Kat, that was a perfect segue into kind of what I wanted to close out with, which is this is a lot of excellent work. But what you started to talk about are, always more open questions, and the very clear open questions that they still have to address with this system. Very interesting ones having to do with what are the receptors for these molecules, what are they activating within the choanoflagellate to trigger these different developmental processes? Is it a stepwise thing? What's the timing of it? So that was a great comment, because you started to kind of touch on building out from this, like where are you going next? And so what I thought I would ask with our remaining time is, so here's what they've done. They knew about RIF1 coming into this. They now know about RIF2. They now know about an inhibitor or a compound that has inhibitory activity. They now know about a synergistic compound, which leads to their very clear title, Bacterial Lipids Activate, Synergize, and Inhibit the Developmental Switch in Choanoflagellates. So I guess I'm kind of curious, like, where do you think all of this is going in terms of a very simple thing that they probably came into this going is that you're just going to find compounds that turn on rosette development. But what they've actually found is this looks like it's far more intricate. So I'm kind of curious where you think this is going in terms of how all of these things interplay as a signal for the choanoflagellate to respond to its environment. So where do you think this is going in terms of trying to figure out the RIFs and the LPEs work together, but you've got this inhibitor that can also kind of reduce rosette development? Where do you think that's going as a kind of a response to environmental conditions for the choanoflagellate itself? And Dave says transcriptome plus CRISPR, obviously. Probably so. Again, if you're HHMI funded, that's perhaps an option for you.
Because that's the answer to everything, transcriptome and CRISPR. No, I like the idea of figuring out not only which pathways which the transcriptome is going to get to and the CRISPR is going to get to, but figuring out are there unique organelles that are triggering this, and then looking at the evolution model, going back to that idea that this is actually an evolutionary model, and going back to the idea of, okay, if we find this system in coagulate, that word, is that something that's conserved all the way to multicellular organisms, and is this a molecule that is important in multicellularity in us or other important vertebrates? I think this is going to be a really fun system to follow, and not just because of the transcriptome and CRISPR that's coming.
Yeah, I agree. Nicole, do you have thoughts on that?
Yes, so I'm going to try to keep this short because my head's kind of exploding because this paper is so awesome. But Definitely transcriptomes for the temporal thing, because part of what I want to know is the timing of releasing all these different factors by the bacteria.
Yep.
So when, what time, and what amounts, how much are they normally secreting anyways? And I mean, the nice thing about this being a cultivatable liquid system is there's some, crude quantitative estimations we can make. It's probably going to be really easy to grind up a bunch of choanos and even possibly use their whole protein as affinity sort of capture to actually find out who's binding all of these. Getting back to the evolutionary component, there's two things that I think are really cool here. The first one is that we've got these cells that kind of do the villi thing that you tend to see in the gut for recruiting, and they're making these stable aggregates that kind of look like an embryo, but they're also forming these surfaces, or I suspect there's probably some type of uniformity of orientation of the villi. They're doing this without a basement membrane. So normally having that layer of collagen down in the gut that you have your cells on. So the other part I'm thinking about is, if this is sort of a precursor system with a minimal toolkit of biomolecules, we've seen a bunch of different sort of long chain lipid conformations with and without double bonds. So number one, these things can probably partition into membranes as well as interacting with receptors, which means that they could also be changing the fluidity of these cell surfaces, further stabilizing or destabilizing where necessary. So the idea of making this multicellular assembly without the need for a proteinaceous base, which is kind of an interesting approach to sort of having, I guess, a limited, a more, well, I'm not going to call it primitive, but a more minimal set of supramolecular structures to work with, let's put it that way. So I think those are also some interesting ideas to kind of look at from possibly even just lipid extracts and looking at, are there more double bonded things? Is this stimulating also different things that have to do with choano lipid repertoires as they assemble these? My thoughts are a bit more disorganized, so I'm going to stop there.
Oh, no, that's okay. I think that's great. I think you're absolutely right. I mean, boy, you could, just in reading this paper, you've identified a ton of things that you can kind of, different directions that you can go into to try to tease out what's going on and then how it applies more broadly. One of the things that Dave mentioned was that he was saying maybe these lipids aren't released or secreted, that maybe they're just kind of slurped off the bacteria. And I think that could be very much the case. And I think these bacteria, as I understand it, many bacteria are kind of constantly blebbing out these outer membrane vesicles. And so it could just be that these are signals that exist in the environment if algoriphagus and choanoflagellates are present in the same area. It could be that these outer membrane vesicles, which they've been using as controls in the experiment, are in fact being released by the bacteria. That's possible too. Dave, did you have any more text-based thoughts, or do you want to make an attempt to see if your internet's improved? It's like, no. Okay, all right. So yeah, so I'm really glad we read this. I'm very much looking forward to hearing more from this lab to try, I'm sure they have a ton of stuff. Oh, wait a minute. Did we just get, Dave just showed us. There's a ton of stuff that I think they're gonna be able to look at with this system. And I actually think that I'm gonna keep this paper in mind, because I think it's, when I teach symbiosis again, I think this is a really good one, because I think it's accessible. I think it's a very clear question that they're trying to address, that they explain really well. And the way they kind of walk through it, this piece, this piece, this piece, add them together, what do you get? Take one away, what do you get? I think it's very accessible for students. So I'm gonna keep this one in mind, because I think it's a really good way of talking about a model system, dissecting that model system, and then trying to figure out how can we learn from that more broadly to think about these big questions that are in other systems. Kat.
Yeah, I just wanted to say that as well. Oh, Kyle has thoughts, it's cute. I just wanted to say I was thinking about that as well, and I thought this would be a good introductory paper for the post-microbe interaction class, in which you introduce a really simple system where you have one microbe and a single cellular organism, and they the signal from the bacteria stimulates this multicellularity in this single-celled organism, and then build up from there. Okay, we have one molecule and one single-celled organism. Let's look at some more complex organisms. So I thought, as I was walking in this morning, I was thinking along the same lines that this would be a really great paper. And I also like the way it's written. I'm one of those people who reads a paper, and if it's not well written, I just get angry. And so I read this paper, I'm like, oh, it just lays everything out so nicely, and I think this would be a really good paper for an undergrad class.
Yeah, and I add.
Yeah. Also, Dave, thanks for sharing us your baby. That's awesome.
Yeah, Dave's son Kyle apparently had some thoughts about the paper, but he's gonna save it until he can like articulate it a little more clearly. Oh, so Dave's saying there's a story of Pseudomonas changing candida morphology by released molecules. So that's kind of a parallel to what we're talking about here. Oh, I'll have to go look for that. I didn't know that. Okay, always back to Pseudomonas, man. Jeez. So yeah, so I agree. I think this is a good paper. I think I'll keep it in mind for classes for sure. And then also just to kind of watch to see what they do with this, because, you know, as we're learning more about host microbiome interactions, you know, this is a great system along with the ones they mentioned, Vibrio fischeri and squid, so forth, to try to kind of give us a handle on more complex systems where there's multiple partners, transient partners, us, our guts, things like that. So thanks to everybody for being here today. I really enjoyed, as always, I really enjoyed talking about this paper with you guys. And hope everybody has a happy holidays. And then we'll probably be back in, well, I plan that we'll be back in January. So if you guys have papers that you are interested in talking about for January, definitely let me know. I'm on Twitter as @microwavessci. So you can tweet at me if you are watching and want to suggest a paper. Or if you guys today want to suggest a paper, feel free to do so. So everybody have a good holiday break, get recharged so that we can come back to it. And Nicole, did you have a thought?
No.
Oh, sorry, you unmuted yourself. So that's my cue to point at people. So I apologize for that. All right, so thanks very much, everybody. And I hope everybody has a good day. It's finally light behind Kat. So I guess it's the sun is rising in Alaska. So the start of a good day. All right, so thanks everybody very much for being here. And we'll see you all in 2017.